Uh, Very excited to present my research to the ISMB community. The fundamental question of my research is to understand how well does a model disease mechanism match the actual human disease. The standard approach to discovering new interventions often starts from preclinical models, such as cell systems and mouse models, and then successful interventions are then translated into human clinical trials. But often, interventions that are successful in preclinical models fail to replicate the success in human clinical trials. So the question is the failure due to the wrong drug, or is it due to choosing the wrong target? Clearly, there's a disconnect between preclinical models and humans, and there are three components to this disconnect. First, models are evaluated based on the proxy of phenotype and how well they recapitulate pathological features of the disease rather than intricate cellular mechanisms. Two, interventions are targeted at single gene targets rather than complex cascades. And three, complex cascades across model systems in humans may recruit varying genes to drive the same pathology. So what if we could represent activity of groups of genes and cascades into one single numbers and pathways? We know that there are inherent noise and subtle changes in gene expression that might not be capturable by standard assays. So if we could look at pathways as units of activity, and if we could determine directionality of functions, whether they are upregulated in the disease or downregulated in the disease, we could form a basis to compare different model systems and data sets from humans. Today I present integrated quantitative pathway analysis that unifies representation by quantifying pathways and selects appropriate pathway within appropriate model to make function portable across model systems and human data. At the core about iQPA is quantification of pathway activity, which, to put it simply, is based on retrieving known pathway gene sets from known data sets, such as M6DB, and masking a given gene expression data by filtering out the genes that belong to a certain pathway and aggregating the activity of those genes into single numbers that represent the activity of pathways. I will not go through the technical details of this pathway activity summarization, but you can see that this is a non-parametric method that is based on ranking the activity of genes. So now I applied this platform to therapeutic target prioritization in Alzheimer's disease. Having activity of pathways quantified, we can perform a series of analyses, including determining what pathways are dysregulated based on linear models, or understanding what are the common pathogenic functions in heterogeneous brain regions of Alzheimer's disease, which we know that are affected differently. So for this question, I analyzed temporal cortex data set from Mayo Clinic cohort of Alzheimer's disease patients and parahippocampal gyrus from Mount Sinai Brain Bank cohort, and compared AD subjects versus control subjects to determine dysregulated pathways. We found that there were 277 pathways that had significant dysregulation common in both brain regions. Interestingly, these pathways had the identical direction of dysregulation. So the pathways that were in common were either upregulated in both brain regions or downregulated in both brain regions. Another question we can answer is basically to understand how similar are different brain regions in terms of AD-associated dysregulation. So for any pathway that we put in iQPA, we can generate a T-score of dysregulation between AD and control. And if we compare this dysregulation T-score between parahippocampal gyrus and temporal cortex, we find a Spearman correlation of 0.8. Whereas if we were to perform the same analysis based on genes, we would get a correlation of 0.5. Now, having this similarity analysis, we can form a basis to extend and analyze multiple cellular models and animal models of Alzheimer's disease. In collaboration with our experimental colleagues at Mass General Hospital, we studied multiple three-dimensional cell cultures of Alzheimer's disease that exhibit varying aspects of Alzheimer's disease pathology. So we compare different combinations of AD lines versus control lines, and we performed an unbiased assessment of functional similarity. So basically, we can get a correlation number between each brain region, represented as brown here, and each cellular model. One of the cellular models that exhibited high levels of toxic amyloid beta 42 positively and significantly correlated with multiple brain regions from Rosemap cohorts, dorsolateral prefrontal cortex, perihippocampal gyrus, and temporal cortex. Using unsupervised correlation, this model system, in fact, grouped with other human brain regions rather than other cellular models. We can go under the hood of this significant correlation to see what pathways are actually driving this correlation. And indeed, there were 21 pathways that had significant dysregulation in AD and control subjects among all three brain regions and our cellular model of interest. To validate this pipeline, we then questioned the activity of P38 MAPK activation, which was the top pathway shared across brain regions in the cellular model. In the amyloid beta 42 high cell line, we first confirmed that the activity of P38 MAPK is increased. Then we performed inhibition of P38 MAPK activity using appropriate inhibitors, and we saw an immediate reduction of phosphorylated tau pathology in P38, and as well as significant reduction of cell death marked by cleaved caspase 3. This analysis can be extended to include other model organisms. So here's a snapshot of including mouse models with cellular models and brain regions. And we can see that iQPA can find significant correlation between different brain regions of mice, mouse models of Alzheimer's disease with human brain regions of Alzheimer's disease. So in conclusion, pathways provide valuable targets for driving translation, and iQPA is a platform to make function portable across model systems and human patients. I would like to acknowledge our colleagues, especially Sang-Soo Kwok, who led this study with me. Thank you.
Thank you very much, Pourya. Questions? Okay. I will start with one. When you were giving the example of the brain and pathways you identified there, there's different cell types and different cells might be the origin of disease or different contributors. Do you think, and gene expression varies between cell types, do you think this needs to be taken into account?
Absolutely. This is an immediate next step for the project, but in the specific case of Alzheimer's disease, we know that neuronal populations are major contributors to the dysregulation profiles that we observe. And this is another aspect without even having the single cell data. Depending on the model system that you use, the cell cultures can include different cell populations. So it can be only neuronal or include neuron and astrocytes. So based on the difference of concordance functions that are recapitulated in different model systems, you can even pin down the functions to specific cell types.
Thank you. Another quick question?
Thank you for walking your talk. Understanding the application of pathways is one standing question. I have just two curiosities whether you use as an output for your model something like a large data set of phosphorylation data, you get that. What is the actual readout of the after the regulation of pathway when you train your model? And what is your plan? I see that you mentioned 3D culture for the testing of organoids and other in-between models.
So yes, there are multiple exciting directions where this project can go. Of course, I think for me, an immediate follow-up is to look into other assays. As you mentioned, this can be phosphorylation assays, proteomic. As long as we have a list of genes and proteins, the method is applicable. And of course, the one aspect that I'm actively working on is to extend to more models. As you mentioned, can be organoid. I just recently included different mouse models, but can be different model systems. So ideally, we would be able to, if you provide a model, I would be able to tell you that what functions, what are the actual functions of the disease that can be studied in this model. So of course, that's a immediate follow-up. Yeah. Thanks.
One quick last question here.
Nice talk. When you did the analysis, did you take care of the age? Because in Alzheimer's patients, you have ages all over, right?
Absolutely. So yes, I tried to squeeze as much as I could in the 7 minutes talk, but absolutely the dysregulated pathways that you see here, because pathways are represented by single numbers, then we can, when we perform linear comparisons between case and control, we can account for all the covariates that can be confounding to the readouts. So in this case, sex, apoegenotypes, age, RNA integrity number, all of these are accounted for in the analysis that's presented here.